Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. We are back today for one of the last pieces of glycolysis, actually. We've hit a large portion of it already. And unlike the last video where we were talking about a relatively obscure enzyme that not many have heard about, today we're going to be talking about pyruvate kinase. And in particular, we're going to be looking at the M2 isoform. And pyruvate kinase has got a lot more kind of information about it. And I think that we have a lot to share. And I think that what was surprising to me when I was going through the medical literature, looking at pyruvate kinase, is that it just reminds me how important each of these pieces are, even by themselves. Because you'll see that pyruvate kinase has diverse biologic effects, and it's a key enzyme within glycolysis to help continue the metabolism of glucose and continue the Warburg effect. But also it's responsible for redox homeostasis and kind of progression and metastases of cancer, and it leads to therapy resistance. So it seems to be a very attractive target for both conventional and unconventional medicine. And I just think that when you look at a very specific piece of the entire puzzle, like pyruvate kinase, and you see such an importance put on it, and then you think, okay, well, what happens if you were to affect multiple steps or multiple pieces in that very critical process? How much better could the results and could the potential be? And I think that's what is the beauty about metabolic therapy, because metabolic therapy for cancer, although, is specific to these processes, it really looks at it from a bigger perspective of glucose as a whole, glutamine as a whole, and lastly, the third part of the three-legged stool, the oxidative stress component as a whole as well. So without further ado, let's get into it. So just to bring us back to the kind of classic graphic that I've been using for kind of giving us an understanding or an orientation about where we're located at. So obviously we started outside the cell looking at blood glucose through the use of a ketogenic diet or exercise or cold or red light therapy or a variety of nutraceuticals, supplements, etc., medications to help lower glucose. We've hit that. We've hit these glucose transporters pretty hard. Then we hit hexokinase. Then we hit phosphofructokinase. Then we hit PFK-FB. Now we're down quite a bit further at another key rate-limiting enzyme called pyruvate kinase. And so, we're basically nearly at the end, because what are we left with at the end of pyruvate kinase? We're left with pyruvate. And pyruvate has one of two major fates. It's going to either go into the mitochondria and participate in the TCA cycle and make energy through OXPHOS, or in the case of cancer, pyruvate is going to be converted to lactate through the enzyme lactate dehydrogenase. And that's what the next and I guess last part of the glycolysis series will be on lactate dehydrogenase. And I'm not sure when I'm going to present that video, just because I have kind of a special treat for us on Monday, talking about, it was motivated by me getting blown up on my YouTube feed, my own personal YouTube feed, about the sugar diet, and seeing how it's kind of trending, and I don't want to say viral, but it's definitely trending for sure. I wanted to address it head on. And although we'll be talking about the sugar diet, it really, the video will end up morphing into a lot more than that. It'll be specifically talking about Warburg metabolism as a whole, but how Warburg metabolism is set up and how glucose will derail any progress for improvement in your situation, and how that worsens outcomes, that leads to therapy resistance, And then I tie back in all of these enzymes and transporters to kind of complete the story. And I think what I initially had planned on doing was I plan on doing a live stream on Monday. And what I may end up doing is I may end up just shooting a video. Right now on the slide deck of that sugar diet slide deck, I made a whole new PowerPoint for it. I think I have like 75 slides. So I think I may just like shoot the video, do minimal editing because I don't have time to really like beautify it too much, drop it Monday. And it'll probably be pretty long because I got a lot to share. I was able to discover a lot. But my point is that metabolic therapy, is hitting glucose as a whole. And I know we're breaking it down, rightfully so, because I think I want to underscore the importance of each of these enzymes or each of these parts that make up the whole. And the whole is critically important, but each of these parts play a very important role by themselves. And I think that hopefully I've illustrated that, where they'll look at like hexokinase and they'll say, oh, if it's upregulated, you'll, XYZ cancers do poorly or have a poor prognosis. Or if phosphofructokinase is upregulated, XYZ cancers have a poor prognosis. And by inhibiting it, it unlocks chemotherapy resistance or whatever, right? And I think that I'd like to show that from a variety of these enzymatic steps to show how important even just one of these steps are. And that hopefully my goal by doing that is underscoring the importance of the whole, but also it also shows that there are very specific important targets that we can look at from treating cancer as a metabolic disease. And I think at this point, considering even a much more obscure enzyme like PFKFB had, many known and a lot of question marked inhibitors, then the more common enzymes that we've seen that we've talked about, such as hexokinase, such as phosphofructokinase, such as pyruvate kinase, such as lactate dehydrogenase, and you've seen the inhibitors that are associated with that, and these are not exhaustive lists of things, hopefully will give us a lot of hope and knowing that we have a lot of tools at our disposal. That's kind of what, one of the goals of the channel is to give you guys at least some degree of hope that there are both natural and drug things that are either already available or are coming down the pike that could be helpful to augment, the foundations of metabolic therapy. So anyway, with that, let's move on to pyruvate kinase. So this paper was published in 2019 and is titled Pyruvate Kinase M2, a multifarious enzyme and non-canonical localization to promote cancer progression. And it says here that rewiring glucose metabolism, termed as Warburg effect or aerobic glycolysis, is a common signature of cancer cells to meet their high energetic and biosynthetic demands of rapid growth and proliferation. Pyruvate kinase M2 isoform, PKM2, is a key player in such metabolic reshuffle, which functions as a rate-limiting glycolytic enzyme in the cytosol of highly proliferative cancer cells. During the recent decades, PKM2 has been extensively studied in non-canonical localizations such as the nucleus, mitochondria, and extracellular secretions, and pertain to a novel biologic functions and tumor progression. Such functions of PKM2 open up a new avenue for cancer researchers. This review summarizes the update functions of PKM2 at various subcellular localizations of cancer cells and draws attention to the localization of PKM2 in the cytosol into the nucleus induced by post-translational modifications. So the bottom line is, although many of these enzymes, and we've seen this with other enzymes too, we've seen it with hexokinase, we've seen it with PFK, we've seen it with PFKFB, we'll see it later with lactate dehydrogenase also. But even though these are critical and rate limiting steps within the breakdown of glucose or glycolysis, or in this case, we're talking about pathologic use of glycolysis called the Warburg effect or aerobic glycolysis. But these enzymes are kind of what they call promiscuous. They have a lot of other roles outside of that. So, and they're not just, they don't have just a role in breaking down glucose. So what this paper is kind of going to unlock in some ways, kind of talk about key important step in the Warburg effect, but also cancer stem cells, angiogenesis, genetic instability, metastases, cancer-associated inflammation, and exocytosis. So in the introduction, just so we all are on the same page about what pyruvate kinase actually is and does. Pyruvate kinase is an evolutionarily conserved metabolic enzyme that catalyzes the irreversible transphosphorylation between phosphoenolpyruvate, PEP, and adenosine diphosphate, ADP, to produce a pyruvate and ATP as the last step of glycolysis. In normal cells, pyruvate is either completely oxidized to CO2 via oxidative phosphorylation, which we talked about. It's going to get brought into the mitochondria via pyruvate, and it's going to be converted to acetyl-CoA via pyruvate dehydrogenase, and it's going to be oxidized to CO2, like it says, in the oxidative phosphorylation system. Thus, well, wait, stand by. I lost it. Which instead, where'd it go? The metabolic reshuffle, now considered as a hallmark of cancer, involves the increased glucose uptake and enormous production of pyruvates, which, instead of entering oxidative phosphorylation, produce lactate without regarding the concentration of oxygen. Thus, cancer cells predominantly confine ATP synthesis to enhanced glycolysis. The phenomenon has been termed as Warburg effect or aerobic glycolysis, described by Otto Warburg. Cancer cells attain such a particular phenotype part by overexpressing glucose transporters and glycolytic enzymes, including pyruvate kinase M2 isoform. One more thing. Although almost all mammalian genomes, including humans, carry two distinct PK genes, PKLR and PKM, which code for the more extensively studied four PK isoforms, PKL, PKR, PKM1, PKM2. The expression of PK isoforms is highly regulated and is tissue specific, entailing that their different genetic properties satisfy metabolic needs of different tissues. So I just wanted to give you that there are multiple isoforms, and I think we'll talk about it a little bit later. A growing body of literature supports the role of PKM2 in regulating redox homeostasis, the adaptation of cancer cells to stress to multiple mechanisms. PKM2 has been found to play a major role in the survival of cancer by shifting the metabolic flux from glycolysis to the pentose phosphate pathway. In response to oxidative stress inducers, hydrogen peroxide mediates the oxidation of PKM2 at this C358, reduces PK activity, and shifts the glycolytic metabolites towards the pentose phosphate pathway. The pentose phosphate pathway plays a crucial role in supporting cancer cell survival and proliferation through the production of reduced form of NADPH needed by glutathione to prevent excess oxidants accumulation. Likewise, the increase in H2O2 or hydrogen peroxide levels enhance SIRT1, SIRT5, and PKM3 interaction, leading to a desuccinylation of PKM3 and the shift of the metabolic flux from glycolysis to the pentose phosphate pathway. On the other hand, enhancing PKM2 enzymatic activity using various activators such as ML285 and DASA10 increases intracellular oxidant accumulation and sensitizes cancer cells to oxidative stress and cell death. So, where did we see this at before? Well, we saw this in a couple of different areas, actually. We saw this in, I want to say we saw this back in phosphofructose kinase. So phosphofructose kinase is this PFK enzyme here. And we saw it act as a metabolic switch to basically determine how much oxidative stress was in the environment and to kind of shift things either towards the PPP, which stands for the pentose phosphate pathway, which is critically important to recycle glutathione and redox homeostasis and cell survival. So this is a really good graphic of this system. So we had seen this when we were talking about PFK and PFKFB, but basically, like, and I was wrong. It's actually this PFKFB that acts as the pair of valves of fine-tuning glucose metabolism, right? And so what happens is glucose is being brought in by the cell by the truckload, 10 to 30x more than a normal cell. But the cancer cell either is going to use glucose to make ATP, energy production, and grow and produce lactate and set up the lactate tumor microenvironment and metastasize, et cetera, right? Or it has to protect itself from the environment. And this is what I call the internal shield system. And NADPH is an important part of that system because it helps recycle glutathione and maintain glutamine redox homeostasis, or the balance of antioxidants and oxidants. Now, this is not the entire story. This is why glutamine is also critically important. We talked about this during the glutamine series because glutathione is made up of glutamate, cysteine, and glycine. So that system is also important with glutamine uptake, glutamine utilization, and the SLC7A11 particular transporter of cysteine into the cell. But it is a system that relies on itself. So like if you have plenty of glutathione available, and that requires glutamine, cysteine, and glycine, but you can't recycle it when it gets oxidized, that's a big problem for us. So these systems are dependent on each other. And so whenever you have, let's say hypothetically, you were doing, hyperbaric oxygen, and you were doing, IV vitamin C or photodynamic therapy or radiation therapy or whatever your oxidative stressor is, right? But you weren't on ketogenic diet, or you weren't blocking some of these pathways so that NADPH could be recycled through the PPP, or you weren't blocking glutamine. You were taking glutamine supplementation. Well, then redox homeostasis will be intact by these cancer cells. They can protect themselves with their internal shield system of this glutathione and other antioxidant systems. So you would be basically like wasting your time and money on things that cost, like the hyperbaric oxygen or the IV vitamin C, or if you were undergoing, conventional chemotherapy or radiation therapy, you would probably also be wasting your time and energy and money, and you'd probably be killing the good cells, and you'd probably be leaving the bad ones intact because they have this shield system, this glutathione shield system. Well, the question is, how does the system know, right, to actually like ramp up this system or ramp up the energy production system? Well, it's based off of, at least from what I've seen, two enzymes now, two systems. The PFKFB system is one way it does it, but also pyruvate kinase. That's what it's saying here. So pyruvate kinase is down here, right? It's right before pyruvate. So pyruvate kinase also acts as a sensor. And what it's saying is that pyruvate kinase is being oxidized by hydrogen peroxide, a key signaling molecule that's also a reactive oxygen species. And when it gets oxidized, it kind of like slows down that enzyme or even shuts it down. So that way, glucose flux is not being shunted towards lactate. It's in pyruvate. It's being it's going to be reversed back to the PPP so that we can make NADPH, so we can recycle glutathione, and hopefully for the cancer cell, what they're thinking is that I want to maintain redox homeostasis. That's what essentially that paper is saying, right in this particular part here. And that is kind of, shown by this graphic here. So when PK activity is low, that means that it's shunting all of the glucose towards antioxidant defense. And if PK M2 is high, that means that it's bringing all the glucose towards lactate and towards pyruvate and energy production, and that leaves kind of this cancer more sensitive to redox stress. So what they're saying is, this is a kind of a confusing graphic, to be honest with you, but as oxidant levels increase, that means that tumor growth is suppressed, as well as resistance to chemotherapy is decreased as well, because a lot of times chemotherapy one of the mechanisms that works is it works upon increasing redox, kind of altering redox homeostasis in favor of killing cancer cells. That's how radiation therapy works. That's how a lot of therapies work. The issue with chemo and radiation is that they're not specific to the tissue. They'll kind of kill everything. Whereas, HBOT and IV vitamin C, they produce the same kind of like effect at the cellular level. It's just that our normal cells actually are either strengthened or not harmed, at least to the degree that they would be by the conventional therapies. But you'll see here that PKM2, although it's a key glycolytic enzyme, and it's definitely a target for us, definitely it also controls more than just glucose utilization. And it's saying here later in the paper, I picked this paper because it's got a lot of good information here. The role of PKM2 in cancer initiation and proliferation. The decrease, sorry, I cannot make the authors, have great English. Unfortunately, a lot of these groups, are from other countries and they don't always. So just bear with me. The decrease in PKM2 expression has been shown to reduce the glycolytic rate and suppress tumor growth in multiple cancer types. So if you decrease the expression of this enzyme, it's going to reduce the glycolytic rate and suppress tumor growth of multiple cancer types, which makes total sense because the hyperutilization of glucose and the Warburg effect is a hallmark of cancer in general, right? So that makes a lot of sense. The reintroduction of PKM2, but not PKM1, restored both the glycolytic and tumor growth, which indicates a critical role for PKM2 in the metabolic shift towards aerobic glycolysis. However, this ultrametabolic state is mostly mediated by interactions of PKM2 with two major transcription factors, HIF1 alpha, which we've talked about over and over again, and c-MYC. PKM2 activates HIF1 to enhance glucose uptake and utilization by increasing the expression of glucose transporters and hexokinase. The activation of hypoxia factor 1 alpha increases the expression of lactate dehydrogenase A and pyruvate dehydrogenase kinase. Pyruvate dehydrogenase kinase, or PDK1, acts as a negative regulator of that phosphorylates and deactivates pyruvate dehydrogenase, which results in the diminishment of the production of acetyl-CoA and the TCA cycle activity. So just so we're all on the same page here. So just pyruvate kinase, when it's overexpressed, okay? When it's overexpressed, it's like a vicious cycle, another vicious cycle. So HIF1 alpha is upregulating all of glycolysis and setting up the Warburg effect. But PKM2, when they overexpress it, when they add it back to these cells, it will auto upregulate HIF1, which upregulates glucose transporters, hexokinase, lactate dehydrogenase, and it also talks about PDK. And that basically shuts down mitochondrial energy metabolism, and it forces upon these cells the aerobic glycolysis and the fermentation of glucose seen in cancer cells. So just PKM2 is also part of the regulation and setting up of the Warburg effect, according to these researchers. I went to the wrong slide. Okay. Finally, c-MYC has been demonstrated to upregulate spliceosomes that demonstrate preference through alternative splicing in the production of PKM2 over PKM1. Taken together, PKM2 regulates a molecular and biochemical network of factors that promote aerobic glycolysis, or the Warburg effect, and cell proliferation. The role of PKM2 in cancer, progression, proliferation, and metastases has been thoroughly studied. Deletion of PKM2, well, I cut that part off. So you can see here in this graphic that PKM2, when the enzymatic activity is low, attenuation of acute kidney, this is looking at multiple diseases, okay? But when you have low activity, that kind of like promotes health, okay? So you're not using a lot of glycolysis. You may be on a kind of more balanced diet or even a ketogenic diet, and that kind of promotes health, right? When as it goes down, it slows down diabetic nephropathy or kidney disease. And then as it goes up, we start having metabolic disorders, inflammation, and cancer. And so, PKM2, although it is an enzyme that breaks down glucose ultimately to pyruvate, it will It also participates in the setting up of various diseases such as insulin resistance, probably non-alcoholic fatty liver disease and fatty liver, other inflammatory conditions, some of which are plaguing our society, like CAD or coronary artery disease, inflammatory bowel diseases. Here's fatty liver disease as well. Steatohepatitis, that's another name for that. And then of course cancer, which is what this video is talking about. Many have also highlighted the role of PKM2 in inducing angiogenesis, or the creation of new blood vessels. Deletion of PKM2 impairs new blood vessel creation or angiogenesis in pancreatic cancer. Conversely, PKM2 released into the circulation positively correlated with angiogenesis in a xenograft model of colon cancer cells. And the intraperitoneal administration of recombinant PKM2 promoted tumor angiogenesis through upregulation of cluster differentiation, CD31, a marker of presence of endothelial cells and tumor angiogenesis. It also promoted migration, cell-to-cell adhesion, cell extracellular matrix adhesion to endothelial cells. So you can see there that this is a big problem for cancer. It's a feed-forward mechanism, because we'll talk about this over a variety of videos, but there are multiple signaling pathways. And as a matter of fact, I talked about it heavily during that last ivermectin video. And remember, that's just the first of 3 videos I'm going to put out on ivermectin. But I talked a lot about the signaling pathways, of how all the signaling pathways will upregulate all of glycolysis and the Warburg effect. And then, as you can see here, sometimes things like PKM2, some enzymes that are upregulated by those signaling pathways are further upregulated by the enzymes themselves, in a kind of a feed-forward positive feedback loop that is really kind of a big vicious cycle. On the other hand, deletion of PKM2 in tongue squamous cell cancer cells lowered the activity of manganese superoxide dismutase, hydrogen peroxide levels, and attenuated cells migration and invasion. These cells were rescued by restoring PKM2 expression, which caused the increase of hydrogen peroxide and superoxide dismutase activity, along with increased cell proliferation, migration, invasion. So the bottom line is, together, these studies highlight the importance of PKM2 in tumor genesis, not just like the energy production that we focus on, especially during this kind of glycolysis system, but also through the entire kind of proliferation and expansion of cancer, metastases of cancer, redox homeostasis of cancer. It's more than just an enzyme that converts one football to the next football, and the glycolysis process. Further on, it talks about PKM2 and the role of it for cancer progression and metastases. Metastases is an important feature of cancer pathology and a major cause of mortality among cancer patients. Metastases is composed of multiple steps, including migration, invasion of cancer cells and surrounding tissues. A growing body of evidence points toward the potential contribution of PKM2 in cancer cell migration. Inhibition of PKM2 suppresses cell migration in gastric and colorectal cancer cells through modulation of PIK3, AKT, mTOR pathways. Further support comes from finding out that diminished PKM2 expression is associated with less invasive TSCC, I'm guessing tongue squamous cell carcinoma. I'm not sure what T stands for, but I'm guessing it's tongue squamous cell carcinoma, compared to that of the more invasive TSCC. Likewise, PKM2 knockout suppresses TSCC metastases to the lung. So there's like a huge, association between migration into other tissues, metastases to other tissues, just by knocking out PKM2, which kind of shows the importance of the enzyme by itself, but also shows the importance of just Warburg metabolism in general is horrible. It at least for cancer, it pretty much like drives the entire process of cancer, immune dysregulation, et cetera, therapy resistance and therapy failure, et cetera. So the clinical significance of targeting PKM2 in cancer therapy: several studies have shown that PKM2 levels in circulation is elevated and could be used as a diagnostic indicator for a variety of cancer cell types. In addition, the overexpression of PKM2 is positively associated with tumor progression due to its role in glycolysis, proliferation, and apoptosis. Recent studies highlighted the correlation between PKM2 activity and chemo resistance in cancer. Specifically, pharmacologic or pharmaceutical inhibition of PKM2 attenuated cisplatin resistance and decreased tumor growth in metastases of bladder cancer. The correlation between PKM2 expression, tumor progression, apoptosis, and chemo resistance make PKM2 a promising target for cancer therapy. So again, we're targeting a very specific thing, and you're seeing the critical importance of one specific thing, but that also highlights the importance of the entire whole. It's really this entire whole is the problem. It's just that we're blocking, we're kind of like slowing the train down, right? By blocking one particular enzyme in this case. Now it gets a little bit interesting here. So insulin signaling and regulation. Insulin is an essential hormone that regulates micronutrient metabolism. In cancer cells, it has been elicited that insulin could favor anabolic pathways and contribute to tumorigenesis by increasing glucose consumption and lactate production. IGF-1, which shares similar activities to insulin, but can also be stimulated by insulin, has been positively associated with several cancers. Treating hepatocellular carcinoma cells with insulin significantly increased the expression of PKM2, while the depletion of PKM2 inhibited the effects of insulin on glucose consumption and lactate production. In a different study, insulin stimulation increased the expression of PKM2 mediated through the PI3K mTOR pathways, which in turn increases the expression of HIF1 alpha. The upregulation of PKM2 seems to play a critical role in the insulin-induced aerobic glycolysis, and the knockout of PKM2 partially inhibited glucose uptake and lactate production. Why do I chuckle? Because it's all connected. It's all connected. And so, I don't know. Let's use this as an insulin receptor or an IGF-1 receptor. It's also looking at an EGFR receptor as well, okay? It doesn't really matter because they all kind of work with the same thing. So basically, when you have hyperinsulinemia, that upregulates several growth pathways, including PI3K, mTOR, HIF1, probably c-MYC as well, multiple other hormone cascades, EGFR, as well as insulin-like growth factor 1. Where do those correspond? Those correspond with upregulating gene expression of all of these glucose transporters and lactate dehydrogenase and PKM2. And then PKM2 and other enzymes can actually feed back and upregulate themselves. So it's kind of like a vicious cycle. So what I'm trying to highlight here is that by being on a high carbohydrate diet, by having high insulin levels, by being pre-diabetic or being diabetic, you are basically set up for failure, okay, for cancer in general. So insulin is a key signaling regulator of PKM2, and then PKM2 also will work through similar pathways that insulin works through to upregulate itself. And what they showed was if they knocked out PKM2 in these cancer cells, like if they knocked it out, at least it partially inhibited the glucose uptake and lactate production, because it's just one piece of the puzzle that is being kind of like downregulated. What we're doing with metabolic therapy is we're trying to hit this from multiple levels. So that way we're not relying on one supplement or one drug or one, therapy to be our savior, right? We want to hit this entire process because we know how critical this process is. And we're seeing that over and over and over again as we look at the little pieces. Just gonna take a look at these comments. How the doctor people are excited for this one. I'm glad you're excited. Hey doc, have you looked much into liposomes? There's a really cool study that shows ginsenoside RG3 from ginseng to make GLUT1 targeting liposomes with incredible results. So I can't say that I've looked at liposomes, particularly for this ginsenoside. I've definitely looked at ginsenoside RG3. I mean, it's in multiple videos. It's part of like the, things that block certain things. I want to say even, not to like give away our PKM3. Here's ginsenoside RG3. It's a PKM2 inhibitor. So I've seen it from multiple videos. So we've definitely looked at it, but not in terms of its liposomal need for administration. I have to be honest with you. This channel a lot of times is looking for proof of concept and connecting dots of how all these systems work together, and kind of give us a roadmap for how to, you know, move forward with what we have in our hands at this time. You know, I don't have the expertise, unfortunately, to look at like drug development, or that's not my area of expertise. You know, I think that this is where I need, like, you know, some of these, like, if any of you guys are pharmacists or you know, biochemists or whatever, you know, profession you would need to be able to, you know, help with that. I think that's where, you know, looking at routes of administration, this actually reminds me of something real fast. And this is not to, you know, not to knock anybody in specific, but you're going to know who you are if you probably hear this. So there's a person who keeps asking a comment, and they keep asking a comment about ivermectin. And they keep asking about, well, if it has effects on glioblastoma multiforme, but it doesn't cross the blood-brain barrier, how can this be helpful? And I've answered the comment multiple times, and I kind of got a little frustrated because I was like, look, I've already answered the comment multiple times. And I basically was like, look, like, I'm showing that there are proof of concept and that it works, right? It's going to be industry. It's going to be other people who design either novel, you know, ivermectins, in this case of, you know, ivermectin that can cross the blood-brain barrier, or other routes of administration such as IV routes or IM or sub-Q or liposomal, in this case, routes of administration that help us get to the serum concentrations we need, or to the target tissue we need to have it be effect. But knowing it has an effect is at least part of the game, right? Because if it has no effect, then just move on, right? Anyway, I'll digress. Another great video. You're welcome. Thank you for explaining. I wasn't able to understand the method and apply this. And maybe I have to wait until the end of the video. Yeah, I think that, you know, what we're doing, again, we're hitting all of glycolysis, and we're hitting and looking at what has been studied that affects these variety of pathways. And today we're talking about PKM2 very specifically. You know, if you go back over the last, you know, probably now a couple months now, we've been looking at glycolysis as a whole, starting with like lowering glucose, blocking glucose transporters, and then looking at certain things that have effects on them, whether it be fenbendazole, whether it be metformin, whether it be, you know, curcumin or quercetin or whatever it is. So that we're doing the same thing with every step or every key step of glycolysis. We can't do every step of glycolysis because I just don't think that all of them are going to be well studied. But the main rate limiting steps like PKM2, definitely we're going to be looking at. With thiamine and its ability to shunt pyruvate to acetyl-CoA, be protective against lactate, promoting cancer in someone that eats carbs and sugar. So Corey, I'm going to talk about that a tiny little bit in the video for Monday when we talk about the sugar diet. I've talked about it plenty of times before. I have a whole video on thiamine and helping prevent pseudohypoxia. I've talked about thiamine in a video. It was a video when I was talking about DCA and having similar effects to that of DCA. You know, there's many things that block PDK. But the thing that's cool about thiamine in particular that is unique to thiamine that DCA and, you know, other PDK inhibitors don't have is that thiamine is actually a cofactor for the pyruvate dehydrogenase complex, or PDH. And so therefore it actually blocks PDK and enhances PDH, which actually shunts glycolysis into the mitochondria. And I've talked about that at length about how, you know, that kind of for some people doesn't make any sense. Like, why would I enhance mitochondrial energy production and energy flux through mitochondria, which we know that are broken in cancer, and we don't want to give them an advantage? Well, what we found, or what others have found that I've highlighted, is that when you put energy flux through a broken mitochondria, you'll radically increase reactive oxygen species, which then unlocks apoptosis, which is blocked. So yeah, your thiamine video is how I found your stuff. Really enjoyed diving in. Yeah, no, thiamine's awesome, actually. The issue with thiamine is that, you know, it's kind of like a double-edged sword. Like if at lower doses, like it's been shown to like kind of progress cancer, I think it's probably overblown, to be honest with you. But, you know, for high doses, quote unquote, and you have to really dig into the literature what high dose means, which generally means 75 or higher times more the RDA, which is not hard to get, you know, but I tend to err when I'm using it with clients. I generally will use much higher doses. Yeah, 800 milligrams is, you're in the by far safe range there. But anyway, so thiamine is pretty awesome. And thiamine, I think, could be very useful in helping with cancer, to be honest with you. Digressing back. So the bottom line is, again, this enzyme is critically important. And although we're going to be looking at several things that can have an effect on it, and I think given that we're already 40 minutes in, I'm probably going to have to do a part 2 of this. But I don't generally talk about, as one of these folks have asked, about routes of administration or dosages on the channel, just unless I'm like really like hammered down and looking at the literature, what it's showing. Because a lot of this stuff, let's just be honest, like is not going to make it to a phase 1, 2, or 3 clinical trial, because there's no money. And let's say, just using the example at hand, there's no real money in taking genosinicide RG3 and putting it through like robust clinical trials, and even if they did, I don't think you're going to have the results you're looking for, because when you look at one piece of this puzzle in isolation, that's how you kind of like, it's almost like it's like a guarantee of promoting the status quo. Because the way I look at cancer is, I look at cancer as a 3-legged stool, right? I talk about this a lot. I look at it as glucose, glutamine, and oxidative stress. So if you're just hammering glucose, like that's why a ketogenic diet in and by itself is not going to be helpful, not helpful, but it's not going to be the answer. It's not the magic bullet. Or if you look at thiamine, or if you look at ginsenoside, or if you look at curcumin or quercetin, you're not going to have the robust effect that you're looking for. Now, some of these things that have multiple effects, like, I don't know, let's say quercetin or EGCG, you may have a better chance because they block glucose and glutamine, and sometimes like artemisinin or other chemicals can actually induce oxidative stress also. So maybe in those kind of cases, you will have more of a robust effect, but you're never going to have the effect you're going to have if you combine it as a whole, right? That's why, metabolic therapy as a whole has to be looked at from those 3 legs. And so I just don't know if we're going to ever have the data. So a lot of times, a lot of this is kind of like speculative, which is why we like are really like hammering down the mechanisms, because if you understand the mechanisms driving these processes, you can understand like where this fits into the piece as a whole, right? This kind of reminds me, I have now been doing these videos for a little over a year. I guess we're now about the 13-month mark, 13, 14 months. And, before that, I was doing a ton of research just on my own, not producing videos, just like putting the story together, right? For months on months, listening to countless Seafried videos and other people's videos, and going through medical literature, setting up this giant PowerPoint, which has evolved into a behemoth, like I think this PowerPoint started out with like 150 slides, and now we're at 1,700 slides. So it's just incredible, kind of like where I've been able to take this information. But my point by saying this is not tooting my own horn. It's that I have exponentially learned over this process, alongside you all, by me kind of having the accountability to present at least once or twice per week with you guys, either in a video format that's produced, which I don't have a lot of time for these days, or these live stream formats, which I have a little bit more time for, but still don't have a lot of time for. But I love it so much, like I really love looking through the medical literature, connecting dots, and then giving that information to you guys as fast as I can. That has allowed me to, be able to take information that I previously had as an integrative doctor, as a functional medicine doctor, as a mitochondriac, as a student of Dr. Cruz, et cetera, as well as Dr. Seafried. That allowed me to, produce a course that I beta tested last year. I think my first kind of beta test course was November of last year. And I had a very small, I think we had like 6 or 8 people in the course. And it was, I have so much fun with these courses, not because I want to see people, in the course because people have cancer if they're in a course, or they have a loved one with cancer in the course. But it's a 6-hour, marathon course where we do, 1.5 hours or 90-minute sessions 4 times. And it ends up me, I have an outline of a course. And I try to make it very, informative, but also practical. And, I had good response, and then I did another one in December and had even a better response. And I became kind of more confident with the material as time went on. And, there's like small evolutions of the course that I've created every month. I try to improve it. And I think the last one I did was in February. And I have not done a course since February, and I'm going to do one in June, towards the end of June. And the course, although it has similarities to the course that I initially had last November, December, because I've learned so much more since then, the course that I'm putting out in June will be, in some ways, a much different course, because I just have so much more information to share. And it's not gonna be longer. It's not gonna be 6 hours of material, but I'm gonna kind of cut some things out and maybe kind of like reorder things in order of importance, because I'm seeing that there are certain interventions that are within the metabolic space that are more important than others, like that have like survival benefit, right? Like not just like this would be theoretically helpful, and this like, this and this. It's more like, okay, when this is studied, like with humans, like people live longer, like 3x, 4x longer. And I've added that in, and I've taken all the information that I've learned in the time being, and doing these deep dives with you all every week. And I've consolidated that into like a new and improved course. So anyway, if anybody has been waiting to take another How to Implement Metabolic Therapy course, that will be coming up towards the end of June. And I would be happy to help you. And even though it is, ask anybody if anybody's been in the course, like it is me, lecturing a lot for the 90 minutes. But what I try to do is, I want everybody to feel like they've got almost like an educational consult, like a personal educational consult throughout the 6 hours, because I leave room for questions throughout the entire series, or throughout the entire lecture. And then I try to, at the end of each like 90-minute session, leave a significant portion of time for questions, because I want people to understand like the material. I want people to have their questions answered specifically for themselves. Obviously, I can't practice medicine, over a course, and I can't be your doctor over a course, but I can answer like the questions to the best of my ability. And I do think this new course I'm going to be putting out in June will be better. Well, I say much better, maybe, but better than what it's been in the past. And I'm really excited to do it, because I really love teaching people and giving people, tools in their tool bag, and also nothing else. It's interesting that the courses end up being like kind of like camaraderie building. Like people like from all over the world are in the course, and they're like, oh, I know I'm from Australia, or I'm from Canada, or whatnot, and people like build like connections that were like completely organic, wasn't even part of the goal of the course. I just want to teach you stuff, but ends up, having almost like a camaraderie that happens. And not to mention, like, we just cover a lot of good stuff, and we uncover a lot of problems, and there's a lot of like recurring problems people have. Like one of the things that I've learned through doing all these consults, and taking care of even patients with this kind of style of medicine, is that, glucose is a huge problem. Getting people's glucose down, getting people's GKI down. And we spend a significant amount of time understanding how to improve your GKI. And now that I've done a whole series on like glucose, a glucose series, not even just like this series talking about pyruvate kinase and very specific enzymes within glycolysis. I'm talking about like how to lower your blood glucose, like how to get your GKI down. Like I've learned pearls over the year, in addition to the, my background in functional and integrative medicine. So anyway, I just wanted to put that out there, that I am going to put a course out. It's been the first time since February, that will help people learn how to implement metabolic therapy. And it'll be based on these principles. And, it's interesting because I think when people take the course, they expect to, have a Seafried course, and you get that. There's no question. It's just that you get more than that also. You get a little bit of me, you get a little bit of Dr. Cruz, you get a little bit of Dr. Seafried, and a lot more. Yeah, the course is badass, actually. I think you have about as much fun as you can have when talking about a horrible disease like cancer, right? Like we really have a good discussion between people. and the issues they're dealing with. And we try to, as a team, overcome the issues they're having, whether it be glucose, whether it be glutamine, whether it be getting substrate, whether it be getting, the right, oxidative therapies, et cetera. And just, it really is good. All right. So we got a lot of questions, comments, 800 milligrams. All I see on social media lately is pro sugar with the Ray P crowd. At first I was like, I'm all on board, but with somebody shouting its benefits, I was a bit like, look, I'm going to completely dissect the sugar diet for general health and for cancer, what it looks like. And then I'm going to systematically dismantle it over a period of 80 slides. Okay. Just wait. I don't think I'm going to make it a live stream because I think it'll be too long, and I'm going to make it a video and I'll publish it. For whatever reason, YouTube tends to like my videos better than it does the live streams. Like I think we get a good amount of people on the live streams right now. We got 44 people, and I'm getting a lot of good comments, and I love responding to comments on the fly instead of having to type stuff after the fact. But for whatever reason, the algorithm prefers the video. So I'm gonna at least try to do one video, 2 videos a month. And the next couple of produced videos, other than the sugar video that I'm gonna produce, will be on ivermectin. That's definitely where the next couple of videos will be on. I clicked the bell on sugar diet video. I'm gonna watch it. So just to be clear, it is scheduled to be live at noon on Monday, but I think I'm gonna cancel the live. I'm just gonna make a video. I'm gonna shoot it, just minimal editing. You're gonna deal with me tripping over words or going off into tangents, and I'm gonna systematically dismantle the sugar diet for cancer, at least. Okay. I'm not saying people can't somehow get ripped on sugar diet. It's just that when it comes to this arena of metabolic health and cancer, I'm gonna give you my honest and usual kind of way about teaching things in gruesome detail about why it's probably not the best idea in the world. So anyway, I'm gonna finish PKM2 probably next Friday on a live stream, but I'm gonna hit this glucose thing as a digression, as a very important digression, because I don't want anybody to chase after the shiny object. Okay, the shiny object right now is the glucose diet or the sugar diet or the high carbohydrate diet. It's paramount that we debunk this, at least for cancer. Okay, I'm not going to talk about it for body composition or bodybuilding or whatever people are doing to get ready for the summer. That's another whole other thing. If you want to have that discussion, do it. If you're 25 years old and you want to get ripped, if there's a way to do it on sugar and it's a better fit for you, go for it. That's not the goal of this video. The goal of this video is to dismantle it as even a thought in your head as being used within the cancer, type of thing. You may have me taking back my bag of oranges I bought this week after the video. Well, Corey, if you don't have cancer, dude, you're probably okay to eat some oranges, That's kind of the point of the video also, is going to be to not a high carbohydrate diet or a diet that is high and not high, but has an abundance of whole fruits and vegetables that have carbohydrates. I don't think is a horrible thing. It's just that within cancer, it's not a good idea. Not for low mTOR, stand MPK. Well, that's where Damien, that's where there's a book by Dr. McCullough that I read years ago called Keto Fast, where they talked about eating very specifically, maybe after a workout to increase mTOR, but then to take advantage of its muscle preserving effects, but then doing fasting for periods of the day where you're having mTOR be low, because we don't want it to be kind of chronically upregulated, especially within cancer. I cook for 2 people in self-help cancer recovery. It's very helpful to understand the science. I'm always trying to figure out what to serve. Well, I'm not a chef, but I can tell you that a high carbohydrate, high sugar diet is probably not going to be the best for anybody. As a cancer survivor, sugar diet scares me, as it should, Amy. My goal is to keep my insulin spikes as low as it could. I agree fully. Thiamine well, as I'll point out, this is my last thing I'll say. As I'll point out, high sugar diets will deplete thiamine, faster than people who are not on high sugar diets. So you definitely have an increased need for thiamine, for sure. I've highlighted that in this video, this upcoming video. I've cut down on fruit and see reduction visually in tumor size. Limit to slight unripe kiwi and grapefruits. Wow. Only beef, meat, eggs, and fish are human foods. Everything else is sales and marketing. I don't know if I feel that strongly, but definitely whole foods. Definitely whole foods. I'm on board with. Anyways, we've gone 55 minutes. We couldn't even get through pyruvate kinase. Let me just do one thing, because there's a lot more with pyruvate kinase. I've lost my spot because I was digressing so hard. Where are we? Right here. We don't have that many slides left of pyruvate kinase, but I will say that the slides we do have are important. They're going to talk about prognosis of people with pyruvate kinase, upregulation in a variety of cancers, as well as therapy resistance, and most importantly, things that can block pyruvate kinase or downregulate pyruvate kinase. So that we will have to finish this up next Friday. But before that will be a detour on the sugar diet. And I hope you all enjoy, because I'm going to put that, I'm still waiting for the neon sign. Me and you both. Watching Sean and Mara show liver visual scans in this one patient, ate a standard diet, had severe osteoarthritis, went carnivore, and mostly healed. I'm not saying that there isn't benefits. I'm just saying that I don't know if I'm like full ready to say that I would do carnivore only. I think there's definitely benefits to plants, is my point. As we can see why a lot of these substances like quercetin or EGCG, obviously these are plant substances, right? So there's got to be some benefits, right? It's just that maybe, not eating all bananas and mangoes would be a good idea for cancer or even for long-term health. Anyways, I'm gonna sign off. Thank you for being here. If you like videos like this, please like, share, and subscribe. If you have not joined the Mitochondriac Army, please consider subscribing. It's free. And anti-inflammatory moves. All right. God bless.